Hi, welcome to today's class. I want to thank our returning subscribers so much. If this is your first time, please click on the subscribe button so you'll be the first to know when I drop another educative video. So for today's class, we're talking on stool analysis. Okay, when the physician requests for stool analysis, usually a sterile container is given to the patient and the patient has to produce the stool sample. Now when the stool sample gets to the lab, the first thing you do is the stool macroscopy. The stool macroscopy. There are several things to look out for in stool macroscopy. You look out for the color of the stool. We check the consistency of the stool sample, and we'll check for the presence or absence of blood and pus. So these are what we look out for. So there are several colors that the stool can actually take up. Usually, we know stool to be brown. So a stool can either be brown, can be yellowish. Now your stool can be green, especially when there's presence of bile. So the presence of bile in the stool makes the stool greenish. The stool can be black, indicating the presence of iron. Maybe you took a lot of blood tabs or these drugs that help to boost your blood level. Can make blood tabs can make your stool black. When you are suffering from ulcer, it can make your stool black as well, because when there is bleeding, when there's bleeding in the stomach or in the body, blood is exposed from the vessel. Now this blood is broken down. What makes the stool black is the blood that is broken down. Fresh blood seen in stool is not does not make the stool black. So it's blood that is broken down inside the body that colors the stool black. So what's the part of the blood that colors the stool black is the iron. You know, blood has the hem and the globin part. The hem is the iron part and the globin, the proteinous part. So it is the iron part, which is the hem, that makes the stool black. So your stool can be made black because of maybe intake of iron tabs or due to ulceration in the stomach or in the body. So your stool can also be bloody. These are just some examples. Also, we have stool that is called rice water. We have stool that is called rice water. Now, rice water stool is seen in patients suffering from cholera. You know the color of water when you wash your rice before you start cooking it. That's your raw rice. Now, when you wash rice, which we eat, when you wash raw rice, that's the color of the water. See that whitish color of the water. That's exactly how rice water stool looks like in case of cholera. So cholera is named rice, gives rice water stool. You see the stool looking like rice water. So that's for cholera. Now let's talk about the consistency. Now for consistency, when your stool takes up a certain shape, it's said to be formed. It's not loosed. It's formed. Then when the stool assumes the shape of the container in which you put it into, usually you're given a sterile plain container. Now if the stool takes up the shape of the container, it's said to be semi-formed. It's not so formed, but it's semi-formed. Now, when the stool is loose, very loose, as in the consistency of water per se, it's said to be watery, especially in case of diarrhea. We're experiencing watery stool. Now, you also check for the presence or absence of blood and pus. You know, I call it mucus. Can call it mucus rather. I can call it mucus. You know, mucus looks like it, it's kind of slimy, like when you're having rhinitis, that's you're having catarrh, the fluid that comes out from your nose. You know how slimy it looks, that phlegm. You see how slimy it looks? That's how mucus. When your stool contains mucus, it begins to be mucoid. It draws like your phlegm now. Then you also check for the presence or absence of blood. Check if there is blood present in the stool sample. So that's how we do our macroscopy. That's for macroscopy. Now move on to the microscopy. Remember, we are analyzing stool. We'll move on to our microscope. For the microscopy, the first thing you want to do is the direct wet preparation. In the direct wet preparation, we use a clean grease-free slide to emulsify aliquot of stool sample using normal saline. So assuming this is your slide, to one end of the slide, you place a drop of normal saline and then an aliquot of stool sample. You emulsify it to obtain a homogeneous mixture. There should be no, you don't need to take so much of the stool sample, just a very little portion. Emulsify it to obtain a suspension. And then to the other end, you add a drop of iodine. And then you emulsify a little quantity of stool sample as well, and you cover both with the cover slip. Now, the iodine part helps you to identify cysts. As some parasites that are seen in form of cyst, like your cyst of Gardia lambia. So the iodine will help you identify parasites that are in cyst form. Now this will help you identify just parasites in the form of egg, that are egg form. So let's assume that you did not see any parasite at this stage, or you did the direct wet mount and you did not see any parasite. You proceed over to the concentration method. The concentration method, we have the flotation method, and we have the sedimentation method. For the purpose of today's class, we'll be performing direct wet mount and flotation method. Now, for the flotation method, we use, we have different types of solutions. We have standard solutions, of which you know the concentration. So in this flotation method, we use brine solution, which is a saturated solution. A saturated solution is one where the solute is more than the solvent to such extent that it precipitates under the solvent. The solvent can no longer dissolve any further solute, so it precipitates. So we use our sodium chloride as our salt to dissolve into distilled water, and we keep dissolving as much salt that will dissolve into this distilled water till it begins to precipitate. And when it does precipitate, we have now made a brine solution. So here we have a brine solution, which are going to use parasites of low molecular weight. Parasites of low molecular weight. It's expected that if they are parasites of low molecular weight, they will float. So what we do, we use a clean test tube. We put a little quantity of brine solution into it, and then we emulsify the stool sample. When we have emulsified the stool sample very well to get a homogeneous mixture, we fill the test tube to the brim. Don't worry, I'm going to perform the practical. We'll fill the test tube to the brim, and once it gets to the brim, we cover with the cover slip and then allow to stand for 30 minutes. Now, you will keep this where there will be no vibration or agitation, because this is going to affect our result. So you leave the test to stand undisturbed for 30 minutes without vibrations or agitation, after which you place it on the cover on this a clean glycerin slide, and then view with the microscope, just as I taught you how to view wet preparations. So that's how we carry out concentration method. And a different day, I'm going to explain the sedimentation method. Okay, so here with me is our stool sample. These are stool sample. Now, let's do the macroscopy together. So if you look at this stool, you see that the stool is brown. The color is brown. Now, the consistency is semi-foam, just like I told you. It's not watery. It did not take the form of water. It's not watery. It's semi-formed. You see, it took the shape of the container. So you say it's semi-formed. This is semi-formed. Now we check. Is there mucus in this stool? I can see mucus. But let me show you. I have to open it for you to see how to detect mucus. You use your applicator stick. Can you see? Can you see that it's slimy? Can you see that it's slimy? So you see these two, it has mucus. So you see it's mucus. So that's how you detect presence of mucus. So now you get a clean grease-free slide. We have a normal saline. You put a drop of normal saline at one end, just like I told you. And then we emulsify. Now, I like picking different parts of the stool sample, just to make sure that if there is something, I'm going to get it. Now we emulsify. Don't emulsify too much of the stool. Don't make your preparation too thick. Just make it light. So when you know you've done a good preparation, this is how it looks. You don't have any solid particle inside. You see this? You see? So you don't have to pack the stool sample. Now we'll cover with our cover slip. Then we'll go over to the second. Now I told you we're making two preparations. I have an iodine to check for presence of cyst. Place a drop of iodine. You pick again. Emulsify. Make sure that there are no clumps. Then we'll cover the cover slip. So now we're going to view both preparation under the microscope. Now I'll take you over to the microscope so you see how it looks like. Alright, so now we are at the microscope and we focus our full sample, direct wet mount. Now, as you can see, what is before you is the iodine preparation. Now I told you that the iodine preparation helps us to identify cysts. So if you look closely at about 1 o'clock, you see two parasites looking round in shape with nucleuses inside. And also, if you look at a little bit away from the center, just opposite those ones, you see nucleus. I think I need to point this for you. That's, if you look, you see this? You see this? This is Entaemoba coli. If you look closely, you see it has nucleus inside. It has nucleus inside. The difference between Entaemoba coli and Entaemoba histolytica is the number of nucleus it has inside the cell. Entaemoba coli has 6 to 8 nucleus inside the cell, while Entaemoba histolytica has about 4 nuclei inside the cell. So you see, this is Entaemoba cyst of Entaemoba coli. You see, this is 1, this is 2. You see, so you see the importance of making both preparations. As if I take it now to the other side, you see that it's difficult for you to identify the cyst, but because of the iodine, it was able to take up the cell was able to take up the iodine preparation and come out this way. Unlike the direct wet mount, which I'm going to show you in a jiffy, you find that it's difficult for you to actually identify it, because this is a cyst, and iodine preparation helps us to identify cyst. So you see the beauty of making both preparations. So now, this is the preparation made with normal saline. If you look closely, if you look at 10 o'clock, you see about 2 there. But because they are cysts, you can't see them so clearly. Because they are cysts. You see the importance of the iodine preparation. You see now, this is the normal saline preparation. You can barely see the cyst there. But if we did not make the iodine preparation now, we won't be able to distinguish. You see how colorless is looking. Especially for cyst, you need the iodine preparation. Now I can see this, although it's not distinct. So that's the importance of making both preparation. If it were an ova or an egg of a parasite, now I would have seen it with this normal saline preparation, but because it's a cyst, it's not clear. So you see the difference. I just wanted to see both preparations and how they look like. All right, so we are done with the direct wet mount. I just want to show you how to do the flotation method. Already, I spoke about it and I told you our solvent is the brine solution, which is saturated salt solution. So now, all we have to do is take a little quantity of the brine solution. You take a little quantity of the brine solution into our test tube. We will not fill it at this point, so we can mix very well. So the next thing is to take an aliquot of the stool sample using our applicator stick. See what I'm doing? You mix till you get a homogeneous mixture. You see that if we had filled the test tube to the brim, we will not be able to mix very well. So you put a little quantity first, and then you mix. Once you have been able to mix properly, that is when you fill the test tube. And remember I said you are going to leave it undisturbed for 30 minutes. So we'll fill now. So I like to make the solution get to the brim so that the cover slip can be in contact with the solution. Don't allow it to overflow. Don't allow the solution to overflow. Just let it get to the brim. So this is the brim. This is the brim. I hope you can see clearly. This is how it's supposed to get. This is the extent it should get to. And then you place a cover slip like this. The cover slip must touch the surface. As you can see, mine is touching the surface of the cover slip. And then you leave it undisturbed for 30 minutes. So start timing for 30 minutes. And once it's 30 minutes, I'm going to show you how it's going to look like under the microscope. All right. So our flotation preparation is up to 30 minutes. So you get a clean grease-free slide. This is a clean grease-free slide. Then you get the cover slip. This is it. You place it on the clean grease-free slide. The part of the cover slip that was touching the solution, you place it on the clean grease-free slide, and then view, just as we did the previous one, using times 40 objective lens. So thank you for following us to this point. And if you have watched this video to this point, why not click on the subscribe button? It doesn't bite. Please click on the subscribe button down below. Share this video with your fellow students. Like, comment, ask your questions. Thank you so much for watching.